Our third speaker today, David Mills with University of California, Davis.
Thank you very much. It's an honor to be here. I will do my best to get through this in time for your cookie break after this. So I'm going to talk about a project that we had looking at the microbiology of food production environments. And I want to start this off by thanking Jonathan for pointing out, just as the last speaker did, that there's a long history here in the microbiology of the built environment, not just necessarily stemming from this project, and particularly in the food industry. They've been studying the built environment, the microbiology of the built environment for a very long time because they worry about pathogen flow, of course, in food production facilities. So this is a critical and essential need. And of course, the new use of omics in this area is really the new thing. But I really respect what John said. There's a lot of data already that exists. So if we think about food production environments, there's different rationales and different thoughts that you have to go through to understand the constraints on those environments. And the two that we've looked at in this particular project are creameries, where they're making cheese, and wineries. And they have very different regulatory needs. They have very different types of equipment, very different operating procedures among the staff. And creameries have very strict codes. You got to wear hairnets. You got to, the floors get washed all the time. There's a lot of sanitation. It's more like a hospital, perhaps. Wineries, on the other hand, are a little looser in their microbiology constraint, in part because ethanol does a wonderful job of eliminating any pathogens from their product. And so I think you might be able to see it here. Wineries have one of the positive shedders that we learned today of microbes. It's not unusual for wineries to have dogs walking around. Of course, they have big garage doors, the air comes in. Very different microbiology situations, certainly environmental situations. And so we were interested in looking at both. The other thing that wineries have that dairies don't have so much are lots of fruit flies. And if you've ever been around a winery during crush, they're everywhere. And we've actually looked at them. I don't have time to talk about it here, but they mirror the microbiota of the winery, not surprisingly. And that's something that I haven't seen around creameries at all. So we talk about the microbiota of the built environment, and there's these wonderful papers that have come out, and people have focused on microbes in indoor environments and tracked where they go. But we're really thinking about a different environment when you're talking about a fermented food facility. You've got things that are going to make a lot of CO2. CO2 is used to monitor for people in those other environments. You can't do that in these environments because you're producing huge volumes of CO2 often in these fermentations and volatile organic carbon. So this is a different situation, and we have to think about it a little bit differently. In this project, we were looking at three different, we had Roger Bolton was a chemical engineer in one of my departments. I'm in two. He's in the Department of Viticulture and Enology. And Kyria Bounty Mills is a yeast taxonomist that led the project. We had actually 3 wineries that we looked at and 2 creameries in Northern California. We took a lot of samples, collected over the 2 years, and we set all the wineries up with the variety of these sensors that I'll talk about for a lot of this. First, I'm going to talk about the sensors. So this was the type of sensors that we set up. They were able to measure VOC, temperature, relative humidity, CO2. There's a particle sensor that we set up in the UC Davis winery. They went to a microcontroller, and then went to a radio module beam to a gateway. And so we could collect data every 15 minutes from many different sites within each winery and each dairy. And if you come to UC Davis, and you go and you look at this little TV screen, big screen TV outside our winery, there's actually live feeds from these real-time sensor data going to this panel. So you look at this panel, and this is a PI core system that was donated to the Robert Mondavi Institute. And there's a variety of built environment projects on here that are live streaming data from the wineries and dairies that were hooked up. So if you click on this one right here, which I think is a creamery, you can actually see the different rooms in the creamery live. And this is actually the past 31 days. You can toggle this. And it's live streaming, VOC, and et cetera on the top. It's hard to describe the impact of this live streaming on the actual participants in this project. So the wineries and dairies we work with each said, can we put some more of these in our other facility or other rooms on top of the project? So we had participants in the project add more sensors just so that they could live stream this stuff, pull it up on their iPhone, and look at their facilities in real time. It's really kind of a fun, it's part of the tour now that they give when people tour our wineries. So these are the 3 wineries. We'll talk a little bit more about the sensor data. We have a teaching winery at UC Davis, and it's novel in the sense that CO2 is removed. Every fermenter has a cap on it. CO2 is piped out. That's unusual. There's not a commercial winery in the world that caps the CO2 and pulls it out of the facility. And we'll talk about that. The other one a winery that I can name anyway is Opus One, which is, those of you who might know, is a rather high-end winery in Napa Valley, and it has a relatively small production volume, and they only make red wine. The other winery we call Winery B is a larger production winery. It's still in Napa, and it makes both red and white wines. So first I'll talk about the UC Davis winery. We had actually profiled this winery from a microbiota standpoint several years earlier. This is a work from a former student of mine, Nick Bokelich. And so we knew that there was a seasonal differentiation to the microbiota of this winery before a single grape comes in, during harvest, and after. So we already had an idea of that, and we profiled this winery in a similar fashion, except in this time we did it with these sensors in. And so we looked at, the sensor mode is a plugged-in wired node here, and satellite nodes in the barrel room outside. There's a winery laboratory here and a classroom that the students used before. Now, remember, UC Davis Winery has CO2 capture. So one of the things that came through with this is you can look at the classroom and look at the CO2 emissions and notice that these happen when they have a class. And that makes sense. And this is 800 ppm or so. Just to give you an idea of a fermentation hall not at UC Davis, here's the amount of CO2 you're dealing with in a fermentation hall during crush, where you're getting up to 4,000. And the OSHA regulations are you can't have more than 5,000 ppm of CO2 per 8 hours, or for 8 hours. So the fans turn on and it blows out when you reach when you get it up to about 4,000. So the Davis winery CO2 actually just sort of goes up and down in sort of the classroom mode because you don't have because the CO2 of course is being piped out of the building. We also had particle sensors in this winery, and we noticed there was a big spike. This happens to be a week in the middle of, or early harvest actually, and we had this big spike in particles. It just so happens that that's actually on a Tuesday, and that was the start of the wine production class. And they open up all the doors, and the dust comes in. And so as grapes are coming in, you can imagine the sudden influx of microbiota that is measured in here. And so it's either that or we have really dusty students because a whole bunch of students come in at the same time. This is actually a picture from that winery. Okay, Winery B is a Napa Valley winery, about 1,000 tons a year, reds and whites. It's got a sort of a standard fermentation hall with stainless steel, a cup, and some wood barrels. It's got concrete floors. It's not a conditioned room. This is a traditional, sort of a typical winery that we see. It's got a barrel room that's stacked high, as you often see in some of these production wineries in Napa. And if you've ever seen them have to fill these barrels, it's rather interesting. They climb all the way up there. And so we put up sensors in this winery. And I just want to walk through some of this data. This is the fermentation hall in that winery from August to mid-September. And you have these big swings in both relative humidity and temperature, and that has to do with when you're opening the doors or closing the doors. You come in the morning, you open up the garage doors, and you open up the doors of the winery, air comes in, of course you're going to have the environmental flux. But the CO2 built up dramatically. And you notice here that you have a buildup of CO2 early on, and it doesn't necessarily go away when they open the doors. It starts to build over the fermentations. And even getting to some pretty high levels, 10,000 ppm. Again, it gets blown out for the most part when you open up the doors. And this happened to be during the early stage of this particular winery's white wine production. They have some early white wine grapes that they crush for early in the season, excuse me. And it's starting to build up here as they go through their traditional fermentation. So we're seeing the activity of the microbes in the winery just from the sensors alone. And it does not permeate throughout this particular facility. This is the barrel room, and it stays at a pretty low CO2 level the whole time. So you're not seeing that CO2 go elsewhere in the winery. Okay, so let's talk about Winery A, and that one happens to be Opus 1. It's in Napa. It's about 500 tons per year, and it makes only red wine. And since I can talk about this, I can mention the gentleman who helped us at Opus 1. He's Michael Salacci. Opus 1 has a beautiful entrance off Highway 29 if you haven't seen it. But the sort of business part of the winery is back here, where they have the fermentation facility. And then a lot of the barrel storage is actually under this portion over here. As grapes go into Opus 1, they actually come in on the second floor. It's called a mezzanine. So they come in on this via an open door that's back here into this mezzanine that has a non-epoxy or non-porous epoxy floor. And they go to a grape sorter here, and it goes up to a destemmer, down to an amazing machine that sorts the perfect berry from the not perfect berry and puffs it out. And then they eventually take it up, gets crushed up here and put into, the juice, or the must, excuse me, gets put into these. And then they position these tanks over these ports in the room. So they just have to sort of move them around, position it, because underneath those ports are the fermenters. And so they drop it down into there. So we also sampled and looked at their barrel rooms. This is a couple of barrel rooms at Opus 1. And as I mentioned before, we definitely see the CO2 rise in the fermentation rooms at Opus 1. The difference and the surprising thing we saw is when you go to the barrel rooms. So here's barrel room 1, and these are the different profiles from June to mid-September in barrel room 1, and all of a sudden you start seeing a CO2 spike in barrel room 1, suggesting that you're actually getting egress of CO2 from the fermentation hall elsewhere in the facility, which we didn't see at the other winery. Now, if you look back at this facility, a lot of the barrel rooms are under here, and so this has to do with the way they think about how to move air around in your building. And when you're in a fermentation production facility that's producing lots of CO2, this is something you want to take into consideration. And so there's certainly nothing, I mean, the levels of CO2 were not high there, but they're witnessable and associated with the fermentation behavior. So we're seeing microbial activity, in a sense, permeate the whole winery. Okay, what about microbiome analysis that we've done? Well, we took a lot of samples, and I'm just going to give a couple of little vignettes. I talked about that floor on the mezzanine at Opus 1 when you bring the grapes in. This is also a place where they take their visitors. So it's part of a tour. It gets lots of people, and it's a gorgeous floor, and it gets cleaned a lot. So there's a lot of attention paid to this floor. And I would contrast that in a sense to a standard, a traditional winery that might have a concrete floor, which is a porous floor, and less human traffic that is of the visitor nature anyway. And so we were looking at the seasonality of the floor here, the mezzanine, and actually the tile floor on the fermentation level below. And we noticed it was quite different. And actually I can just show it better in these plots, in these ordination plots. And you can definitely see a seasonality before, during, and after crush to the floors in Opus 1. And we've seen this before to the floors at UC Davis Winery. The difference was in the other winery, Winery B, which was more traditional sort of winery setup and had a concrete floor, we didn't see much change throughout the seasons. So the choice of flooring might influence the persistence microbiota in that facility. And remember, we're bringing a lot of microbes in. I expect to see seasonal differences in these floors. Okay, what about creameries? We've actually profiled a couple of creameries previously. Again, this is the same student, Nick Boklich, from a couple of years ago, profiling organic artisan creamery in Northern California. And this is another heat map of which organisms are showing up where in this creamery. In this case, we just took one sample. We didn't do time course. And I wanted to point out a room here, or an area here, that was a Debaromyces. At the time, we typed it as Fabri. We've since typed it as Hansenii. That was in the Bloomy Rhine aging room. So that's this room over here. And in this kind of cheese making, here it is, sorry. In the Bloomy room, aging room, we noticed a lot of this species, Debaromyces species. And this makes sense because you go from fermented curd, essentially, to this beautiful white furry Debaromyces growth on the surface of the cheese during this aging. When we actually looked at the sensors for those rooms, it's really got quite interesting. So once again, I'm talking about these rooms over here. Here's the sensor plot for several months, and you can actually see their cheese production facility, or their pattern over this time. So you see CO2 spikes and VOC spikes that are related to that growth of the Debaromyces in that room. And then you can break it down a little smaller, and you can see the periodicity. As they move new fermented cheese into the room, it starts to grow the Debaromyces on the surface, and then they move it out. Again, the cheesemaker was fascinated with the sensor data because it helped him understand his biological process. And you wonder if he can start making movement decisions, logistic decisions within his cheese facility on the basis of real-time data like this that he's getting back. So it's part of the rationale when he came up and asked for, can we put more of these sensors in? So we are still going through a lot of the microbiome data. There's all these individual stories, and we're trying to sort out which ones to put in what paper. So that's ongoing. And to summarize, I would say that the real success of this story, I mean, the breakout success from the stakeholder perspective has been the sensors. They're really fascinated with these sensors, and every time we've put them in a winery, people have wanted to expand it. As a matter of fact, we tried to talk some of the students that there might be a company if they wanted to start it, and they smiled and nodded and looked at us and then went to work for Google. So we sequenced a lot of samples. I think following VOC and CO2 shifts in these kind of facilities become quite useful, and this kind of data certainly can be used to follow spoilage. I want to get back to one last point before you have your coffee, and that was something that Paula said at the start of today. Will scientists, engineers, architects, and practitioners collaborate? Well, first and foremost, I'm from a food science department, so other than architects, we already have all that group in our average faculty meeting. I don't know if you want to call our faculty meetings collaborations, but that is an average day in our department, so that's normal. But I was talking to Roger Bolton, who's one of the other leads on this project, and he's really an eminent scholar to the wine industry. And he's using this data to go out there and sell a new design of a winery, and saying that here is the first real-time evidence, lots of data collected to help wineries design a new type of winery that can be more sustainable and more safe. And so this is working, and these kind of data are getting out to the stakeholders and changing their practices. With that, I'd like to acknowledge all the collaborators, particularly Roger, Andre Knudsen, and my wife, Kyria, who led the project, and a lot of students who took a lot of samples. Thank you very much.
Time for questions?
No, I don't have any wine.